Hi and welcome to my poster presentation. I'm Lara, doctoral candidate 10 from the MSCA project ViroiDoc, and this is a poster presentation or concept presentation of my PhD project on RNA sprays, precision tools for the modulation of host genes to control viroid infections. As you may know, viroids are the smallest plant pathogens, but they pose a big threat to global agriculture by causing severe yield and quality losses. Besides eradication measures, there's currently no proper sustainable way to control viroid infections. In my working group at the University of Regensburg in Germany, we work on spray-induced gene silencing, which could also be used against viroids. Spray-induced gene silencing is a technique based on RNA interference. You basically spray, for example, double-stranded RNA targeting a pathogen on top of a plant. The plant takes up this dsRNA, and dicer or dicer-like enzymes can cut it into small interfering RNAs. These get incorporated into the RISC complex, where argonode proteins cut complementary mRNAs and thereby induce gene silencing. In my project, I will use this and develop different kinds of RNA sprays to either directly target viroids or boost the host plant's defense. I will mostly work with hop-stunned viroid, citrus bark cracking viroid, and hop-latent viroid in hops. To develop RNA sprays, I will firstly need to identify viroid and host targets. For this, I will do RNA sequencing of infected and healthy leaves to identify viroid-derived small RNAs, or use already existing datasets. With these datasets, I can also do in silico target prediction to see which host genes are targeted by viroid-derived small RNAs. Another method I might use is GPMS, which stands for Comprehensive Identification of RNA-Binding Proteins by Mass Spectrometry. Here I take viroid-infected plants, add biotinylated probes that bind the viroid RNA, capture them with streptavidin beads, elute the bound proteins, and then identify them by mass spectrometry. This way I can find host proteins that bind directly to viroid RNAs. These proteins can then be used as targets for EpiEdit. EpiEdit is based on a dead Cas9 enzyme, dCas9, which is guided to DNA by an RNA. Instead of cutting like we know it from CRISPR-Cas9, dCas9 is fused to an epigenetic modifier. This modifier can either methylate, demethylate, or acetylate DNA or histones, which either positively or negatively regulate gene expression. With this technique, we can enhance host defense or suppress proteins that are needed for viroid infection. In the third part of my project, I will design different RNA sprays, like for example, double-stranded RNAs. Double-stranded RNAs could silence known host factors like the transcription factor 3A, 7ZF, or VIP1. Or they could act as decoys by binding to proteins like RPL5 so that the viroid cannot bind to it. Small RNA sprays work similar to double-stranded RNA sprays. They could be used to neutralize viroid-derived small interfering RNAs. Circular RNAs could be designed as antisense RNAs or as RNA sponges with multiple binding sites to trap the viroid-derived small RNAs. mRNA sprays could express, for example, nucleases like HBN1, which are translated in the host and cut viroid RNAs. Finally, all of these sprays will be tested on plants and their efficiency will be evaluated. Please check out the ViroiDoc website or the Koch lab in Regensburg by scanning these QR codes. Thank you.